We have done this, but let's do it again. We are again talking about the different pressures at different levels. I am doing this for the third time. It is so important. What are we doing? Pressures. Where? At different level, at different stage as the blood flows. Let's go there. Renal artery, 100. Okay. Afferent arteriole, 85. Glomerular capillaries, 60 and 15, and we have done it. Are you okay so far? Now, by the time the blood goes into this capillary network, this also we did today, it's about how much? 18 to 8. By when, as it enters into this capillary network, the pressure is about 18 mmHg, and by the time it is ready to enter into the veins, it has fallen to about how much? You know something? Here follow this. Starling hypothesis is operating on a larger scale. I'll explain to you. But Starling hypothesis, what was Starling hypothesis? That tiny 1 mm long capillary, okay? On the earlier part, on the arterial part, the fluids come out, and in the venous part, the fluids go back. Hello? And whatever doesn't go back is the lymph. We did it. Yes or no? Hello? Are you okay so far? Now, here a very similar phenomena is operating, but on a larger scale. Lot of ultrafiltration is happening here, okay. The blood as it goes eventually, I'll ask you a simple question. I'll collect blood from the afferent arteriole and I'll collect blood from the efferent arteriole, okay, 100 microliters each, and I'll subject both the samples for finding out what is the amount of protein in it, which is going to be more and which is going to be less. Tell me, did you follow the question? Okay, I'll draw the blood from here. Are you okay? How much? 100 microliters. And then from here, 100 microliters. The two samples, and I'll subject them to analysis just to find out what is the total protein content in both. My question to you is the protein content in both my samples same? A is more or B is more? They are same.
Concentration would increase, but the content would be.
Concentration, content I am asking. Content would be? How it would be same?
It will decrease.
How it would be same?
Protein content will still be same to the smaller protein.
Yeah, that will.
Okay, let's see one thing.
There is a filtration is happening. Okay, lot of plasma has gone.
But large proteins are still there. So when I go to the efferent end, okay, the blood is actually more concentrated. Hello, plasma has gone. 20% plasma has gone. Okay, but the proteins are still there. So in the 100 microliter of sample, I am of course going to get more proteins.
Hello. Hello. You're not listening. Okay.
Okay, so we are having plasma, cutting certain proteins. We are entering into the glomerulus, and the plasma goes away. What about the proteins? They are still there. Okay, so the number of proteins molecules per unit volume is more. Okay, and that's what I am going to estimate when I collect the sample from the efferent. Now, as a result of the beauty of physiology, as a result of more protein concentration in the blood that is flowing through the peritubular capillaries, because there is more protein, they exert more oncotic pressure or osmotic pressure, you are right, and the result of that they facilitated, and the pressure itself is less. Pressure is less. The pressure is how much? The pressure is how much? Maybe 8, 10. Pressure is low. Because the pressure is low, okay, it will facilitate the influx. Because the proteins are more, it will also promote the influx. So the peritubular capillary network makes excellent ambience for the reabsorption of lot of filtered material from the nephron. You want to take back a lot of water, you want to take back of glucose, this is being facilitated by, particularly for water, number one, oncotic pressure has gone up and blood pressure has gone down. It will facilitate too. And so in a way, this is like, again, catch the point, this is like earlier part of the capillary, whereas this is like the latter part of the capillary, where the Starling hypothesis is operating on the, I think I have done enough, let's move on. So, kidney is such an important organ, and blood pressure at which it operates is so important that the kidney has said that I don't really want to depend on the blood, I want to free myself from the burden of blood pressure changes anywhere else in the body. I am going to make sure that the blood that flows through me is kept at a constant pressure. You know what they did? That kidney has almost total, not almost but quite total autoregulatory mechanism, autoregulatory control itself with blood pressure was demonstrated in a very simple way. A little cruel, but a killer dog collect its kidney, perfuse the kidney. Okay, what do I mean by perfuse the kidney? You have separated the kidney. Just look at the experiment, you know, physiology people do amazing things. Number one, you have 3 cannula, plastic cannula, okay, of the proper size. One cannula you insert into the artery, hello, one cannula you insert into the vein, and third cannula you insert, which is a thinner diameter, you insert it in the ureter. So, for all practical purposes, the kidney doesn't know that it is outside. What a funny language. Kidney knows, it doesn't know. Kidney is there, okay. And now you remove the blood, and instead of that you start perfusing, use the language, perfusing the kidney with physiological solution in which the pH is proper, in the osmolality is proper, everything is physiological. So, actually what is happening is that fluid is getting ultrafiltered, okay, some of it is going in the form of urine, some of it is coming back via the vein, and now you do something interesting. When you begin the experiment, you start pumping in the fluid. I am just taking for example, you are pumping in the fluid via the renal artery, you are pumping in the fluid 60 mm Hg. What do? 60 mm Hg, 60 mm Hg, good. And then you keep on monitoring the urine, it's not urine, it's just filter, the urine output or whatever output you are getting through the ureter. Are you with me? Experiment, look at, imagine the kidney, 3 tubes, one takes the fluid in, another takes the fluid out, and third collects the filtrate. Done? Now you start with applying the pressure into the renal artery 60 mm Hg. You get fluid filtering in the ureter at a particular rate. Good. Now increase the pressure from 60 to 70, 60 to 70, 70 to 80, 80 to 90. You find that the rate at which the ureter is discharging the fluid is still same. Hello, something is happening, something very interesting is happening. Means what? The kidney is capable of, or if you are in that scenario, okay, let's do something simple. In that scenario, you insert your the fine capillaries, okay, to find out as to what is the pressure at which the ultrafiltration is happening. It is still about same. It is same. The kidney is able to adjust, and that is shown in this diagram. Look at this graph. You are increasing the blood pressure from okay, the mean is about how much normal, 80, 120 is about 100. Hello, are you okay so far? So I am what point? I am at this point. Okay, and then I can increase the pressure of 100, 150, or I can go to 100. That's very high actually. Okay, this guy is still alive, but he is still okay. Okay, but in this range if you see, the glomerular filtration rate is almost stable. Can you see that stability? Appreciate the point. What is the message here? The message here is that although the blood pressure can go elsewhere in the body, can go from 100 to 20, 150, okay, the glomerular filtration is same. Okay, how is, and don't forget, this is an isolated preparation. There is nothing coming from autonomous nervous system, no sympathetic, no parasympathetic, no hormone coming from somewhere. This is the kidney on its own. Are you with me? What is the point that author wants to hammer here is that kidneys, the ultrafiltration that is happening in the kidney is so important that the pressure at which the pressure has to be kept constant, and for that the kidney has auto-regulatory mechanisms. I put this slide and explained the phenomena and explained the experiment only to emphasize the point that the kidney has the capacity to maintain the pressure. Now, most exciting question, How does kidney do that? Okay, all right. One is of course angiotensin, as we said, but I'll come to that a little later. In the literature you will find there are the authors have come up with two different explanations. They are not mutually exclusive, so maybe both are right, whatever, but let us study both. One mechanism which operates is called as myogenic mechanism. What is it called as? Okay, myogenic mechanism is muscle. Hello, we are talking about muscle. What are we talking about? Well, here the point is pretty simple. He says that if the blood that is entering into the Bowman's capsule, suddenly pressure goes up. If the pressure suddenly goes up, then look at that inlet and look at that outlet. Are you with me? Afferent and efferent. As a result of that, the afferent vessels will stretch. Hello, they will stretch. But then don't forget the basic physiological property of a muscle. If you stretch it, it will go back. Okay, and here it goes back a little more. That is the property of the muscle, and as a result of that, this afferent tube will constrict. Once it constricts, the pressure is not the high pressure there is not transferred to the inside. Are you with me? Means the pressure is more, but you have reduced the diameter of the tube. As a result of that, the rate at which the pressure at which the ultrafiltration is happening is now reduced. Are you with me? Hello? Am I making sense or not? Hello? No? Do you want me to do it again? Oh, great. Let's do it again. No, this is important. Okay, let's simple. Let's forget all this. I'll take a simple tube. Take a simple rubber tube. Okay, and the water is flowing through it at the rate of 60 mmHg. That is the pressure. Okay. Then artificially I increased it from 60 to 80. As a result of that, the tube has now what? Swollen. Okay, because there is more pressure. Of course, it will stretch. Okay, but my tube is a magical tube. It's a magical tube, and by magic I have told the tube that if somebody stretches you, you do just opposite. Are you getting that message now? Just do just opposite. It's muscle. It's a smooth muscle. Okay, and the smooth muscle has a property. If you stretch it, it goes back. Okay, and because it goes back, it not only means it has gone back. So it was originally so much. Because of the pressure, it becomes so much. Now, if it remains so much, it will take the pressure of 80 mmHg to the glomerulus. No, because it goes back again. It goes back. The fluid is going back into the kidney again at the same pressure of 60 mmHg. What do we call this as? Are you okay now so far? Hello, everybody. Okay.
A little confused.
Sure.
When it contracts, the afferent pressure, the pressure in the afferent artery will increase, right?
It will increase, but just look at it, the amount of the inflow will be greatly reduced. That will really material influence the, okay. Yes.
Not have the opposite reaction, right? If the pressure decreases.
That is the property of this. That is the property of all smooth muscles. Okay, you stretch them, they'll go back. If you want to keep them stretched, okay, you have to instruct them accordingly. Okay, or if you want to keep them relaxed, or
if we don't stretch them, if we decrease the pressure, they don't do anything.
Oh, they respond to that also.
So they expand.
Yeah, they respond. They will. If you mean to say if you are reducing the pressure, if you reduce the pressure, they will again constrict. They will. For that you have another mechanism. I'll tell you. Yeah, I'll come to that. Okay, yeah.
In the previous slide, if we increase the pressure, will ultrafiltration rate also increase?
If you increase the pressure, the rate of ultrafiltration will also increase. That is the problem. Okay. We have already seen, the effective pressure was 10 mmHg. Okay, if you make that 60-70, that effective pressure will be more. Okay, that 10 may become 20. I don't know. It may become 18. Whatever it will become. Okay, we don't want that to happen. For the kidney to function efficiently, these are the optimized parameters, and you can't mess up with them. And therefore, kidney insists that the kidney gets the input at 60 mmHg. But okay. Now, whereas this is myogenic mechanism, I will tell you about another now. Hello, wake up everybody. Wake up everybody, something very interesting is going to happen. Wake up. Are you ready? Oh, this is going to be an interesting ride. Be with me. This mechanism is called as tubuloglomerular feedback. If you sleep, you will not follow. Wake up. What am I talking about? Tubuloglomerular feedback. In this again, let us imagine for some reason, okay, the blood pressure has gone up. Okay, and for some reason the myogenic system did not work. Imagine what is there. It didn't work. So now more blood is going into the glomerular setup. Higher pressure. Higher pressure. Higher pressure means more filtrate. Hello. More filtrate. More filtrate means more everything, including more sodium. Hello. So suddenly now in the tubule you have, okay, I don't know much, but I suddenly experienced that there is more sodium. Hello. In the tubule there is what? There is more sodium. So where am I? Okay, I am here. There is the filtrate is happening, the filtration is happening at a particular rate. Are you okay so far? Good. Now pressure has gone up. As a result of that, the filtrate is more, and as a result of that, in the filtrate that is going on, or the pre-urine that is going, is now consisting of more of everything, including sodium ions. Now the sodium ions and everything goes here, and here we have already said that the distal convoluted tubule talks to the Bowman's capsule. You remember that rule? Are you okay with me? What? The distal convoluted tubule of the same nephron is going and talking to what? The same Bowman's capsule. Okay. Now what I will do is, I will now magnify, I will take this tiny part and blow it up to make my story simple and interesting. So where is the urine passing? The urine is passing through this tubule, and it is lined by a single layer of cells, epithelial cells of the nephron. Are you okay so far? And on this cell, the single layer of cells, I will call this cell which comes in contact, which directly comes into contact with urine under formation. It is not formed yet. I will call this as the apical side, and I will call this as the basal side, and this is of the tubule. But on the other side there is that Bowman's capsule. Are you with me? That Bowman's capsule, and in the Bowman's capsule you are having the afferent input, so that afferent arteriole is going this way. Are you with me? What am I talking about? I am talking about the cells of the urinary tubule, and the urine is going this way. On the apical side there is a protein system. Very interesting. I will call that protein. Look what is written there. I will call it as NKCC system. What do I call it as? Why do I call it as NKCC? Sodium, potassium. So CC, NKCC. Okay. Now I am telling you NKCC because this NKCC term is going to come 100 times. NKC, it's a system of proteins. It is a system of proteins which is a symport system. That symport system is driven by sodium ions. Why sodium ions? Because sodium ions would always like to go from outside to inside. 142 times more. Hello. Sodium is for 140 times more as compared to 10 times more. I'm sorry about that. 10 times more than that. So sodium, and then so that drives the chloride and potassium. So all those things are. So now suddenly those cells what? The cells which are lining this. Can you see this dark patch here? That dark patch is these cells. Okay, and that dark patch of cells, these cells, is called as macula densa. What do you call it as? These are the cells of the urinary tubule. Cells of what? Urinary tubule. Distal. So the Bowman's capsule, proximal convoluted tubule, loop of Henle comes up, talks to the Bowman's capsule. This tubule has certain cells which I am going to call as what? So those reddish cells are all the cells of macula densa. And the cells of the macula densa on their apical side, in their plasma membrane, they have a protein system which I will call as what? NKCC, and that facilitates the transport of sodium ions from the extracellular medium within the cell. Within the cell now suddenly you have more sodium ions. As a result of more sodium ions, the osmolality goes up. As the osmolality goes up, it draws more water from outside, and as the more water enters into the cell what? The cell swells. And as the cell swells, the cell is equipped with stretch activated ion channels. Okay, and as a result of that, two interesting things happen. Number one, the ATP escapes from the cell to the outside, okay, and then the ATP escapes from the cell, and then the ADP also comes out of the cell. We have done it actually in one of the lectures, if you remember. It is converted into adenosine. We have done it in the case of heart, adenosine A1, A2 receptors, yeah, and then it shows this ADP converts in adenosine. And what are these? These are this is the afferent blood vessel. The afferent blood vessel just outside has smooth muscles. Those smooth muscles are equipped with receptors for adenosine. And can somebody tell me what is P stand for? Very good, purinergic. Very good. What do you call them as? Purinergic receptors. Well, ATP, look. I am not looking at ATP as an energy giving molecule. No, I am looking at ATP as a signaling molecule. And if it combines with its receptor, I am going to call that receptor as a purinergic receptor. And that purinergic receptor is sitting on the plasma membrane of the smooth muscle cells. And those smooth muscle cells are going around the afferent arteriole. And when they receive the signal in either in the form of adenosine or in the form of ATP, they will what? Constrict and reduce the blood supply. Hello? Are you okay? You want me to redo this? Okay, I'll do it. Yeah, depending. Oh yeah, we have enough time. Okay, let's see. Do it again, no problem. This has to be understood. No, actually this has to be, yeah, no, enjoyed. Have fun, okay. I mean, this is the beauty of biology as to how the system takes signal from something that is going wrong, okay, and take the signal to the right target and give the correction, give the proper correction. In this particular case, what has happened is for some reason the blood that is entering the kidney, it should enter at a particular rate that has gone up. As a result of the filtration has gone up. As a result of that, one of the products of filtration, which is sodium ion concentration, has gone up. Number of sodium ions in the filtrate are more. Okay, they were sodium ions were so many sodium ions were there, now suddenly 10% increase in the number of sodium ions. And those sodium ions will go all the way down to the tube loop of, and they will come up. And there's now the medulla densa is only there, not everywhere. They'll talk to the medulla densa. Medulla densa, no, what the medulla says, are the medulla sense? Can I not say that the cells of medulla densa are sensitive to sodium ion concentration? Hello? I can say that. They are sensitive to what? Sodium. And how do they respond? They respond because when there is more sodium, they just take in the sodium. I mean, they don't know that this is Na+, okay? No. They don't know that this is Na+. No, they don't know that. Okay? They know that something has come in. As a result of that, osmolality has gone up. That also they don't know. That also they don't know. Then as a result of increase in osmolality, there is more water coming in. And when water coming, it stretches. It is the stretch which the cell knows. Okay? Because it has stretch receptors. And then it responds. Okay? That's a signal that is receiving. What is the signal? It has stretched. Okay? And then it gives out a signal. It's a chemical signal in the form of what and what signaling molecules? ATP and ADP. Okay, that ADP is converted in the outside, it is converted into adenosine. Okay, and adenosine combines with a type of receptor called as A1 receptor. Where is that receptor? Sitting on the smooth muscle. Okay, the signal will go in. Okay, it will bring about the cascade of events. Calcium ion concentration will go up. Okay, calcium ions are suddenly available for actin myosin filaments to slide in that smooth muscle, and the smooth muscle will constrict, and the inflow of blood in that particular nephron will be restricted. Are you okay so far? Okay, just appreciate the beauty of the physiological mechanism, okay, which the kidney has evolved so as to ensure that the blood flow. Why are we doing all this? We want to make sure, one second, we want to make sure that the ultrafiltration keeps on happening at a steady rate, no matter what happens outside. The heart may go up and down, there may be changes elsewhere, but as far as kidney is concerned, the kidney wants to make sure that the ultrafiltration. Yeah, go ahead with your question.
Here it shows that the
macular denser cells or the tubule interacts with the capillaries, not the.
No, on the capillary here. This is not a capillary. This is the,
Yeah, but on that, because it's, isn't it inside? Like, isn't that the level of capillaries at the?
here you mean?
Yeah.
Talk to you afterwards. Don't take the time of your entire class. Huh?
How does the ATP, it just diffuses out or like how does it?
Oh, okay. There are a range of proteins. At this moment, I remember the name of one protein system called as pannexin. Pannexin, we have encountered this once. So if you go to earlier slides, it's there. It is such proteins which allow these molecules just don't go. There are channels. Okay, it is through those channels that the ADP or ATP they can flow out, and then eventually ATP can directly find its purine receptor or adenosine in the cytoplasm. If you go to your slide on heart, you will find how ATP is converted to ADP, AMP, and finally adenosine. Okay, and that all happens in the extracellular matrix. Okay, and but your question is correct. How does ATP go? It goes via certain protein channels, and I remember the name of one protein which is pannexin. Yeah, somebody had a question here.
It's the same question. So the pannexin opens up during stretch.
Yeah. Exactly. Lot of changes happen. Okay, and one of them is opening of pannexin and allowing the ATP molecules to go out. I mean, means you see, you are that's a beautiful question, but then we actually I am not answering your question, but I'll paint the entire scenario. You find out, okay, the cell has stretched. So what? What next? So there are several steps. Okay, and then somewhere in the end you will find that pannexin, the channel open, and then ATP goes out and the rest of the things. Okay. Yes. Okay, solved. Okay. All right. Yeah. Anything else? Yeah, please.
After the smooth muscle contracts in the afferent arteriole, the rate of blood flow through the afferent arteriole.
Yes.
How does that also reduce the GFR?
Okay, I'll ask you one question. Supposing the afferent arteriole had not constricted, what would happen?
Used to have increased blood flow rate at higher pressure.
Higher pressure. So from 60, you might have gone to 80. Okay. Now by this mechanism, you have been able to bring it back from 80 to 60. Makes sense, huh? Okay. If you had not done that, just imagine what would have happened. Okay. All right. Anything? Yeah.
If we consume more amount of salt, like you said, so that it will be less reabsorbed.
Which one?
Like when you said, okay, if we eat high amount of salt, say maybe increase the maybe osmolarity of salt in the blood, then more salt will be filtered and less of it will be reabsorbed. So the
yes.
will have more salt.
Yes.
Even then, technically, this should be like this mechanism should be triggered even then, right? And because of that, the glomerular filtration rate would.
See, the answer to the question is, I'll make a funny statement. Heart is an endocrine organ. What did I say? If I make that statement, I am answering your question, okay? Sounds absurd, but it is not. Heart is an endocrine organ. Okay? Then you can ask me, what is the hormone? The hormone is a protein. Okay? It is called as atrial natriuretic peptide, ANP. I am sure some of you have heard. What do you call it as? Atrium. So the cardiac muscle cells are serving as a hormone. Natriuretic, losing sodium ion, getting rid of sodium ion. So when you have more salt intake, that system gets activated, okay, and that acts on the kidney and facilitates the excretion of sodium. We'll talk about it when we talk about the how the sodium homeostasis is maintained.
Even in that case, the sodium will be more in the filtrate.
Yes, it will be more.
Doesn't that trigger?
No. Let's see it in that light. Don't go to that question now. Okay. My argument is, you cannot take only this part of the solution and talk about that problem. No. If you want to understand how the excess of sodium is being dealt by your physiology, let us take this picture and let us see how what excites ANP and what is the role of ANP in regulating the kidney, and then let us take a look at it in totality. Just don't go for this. I can't answer this in this fraction. I can answer it in totality, but I don't want to do it now. Do it when we do atrial natriuretic peptide. But that good. It gives me an opportunity to tell you. So what is heart? It's also an. And what is the name of the hormone? Atrial natriuretic means excretion of urine peptide. Okay, about 27 amino acids, if I remember.